I'm Bill Hoffman, Communications, Department of Laboratory Medicine and Pathology. I'm here with Dr. Leo Furcht. Dr. Furcht is the Allen-Pardee Professor of Cancer Biology and Head of the Department of Laboratory Medicine and Pathology at the University of Minnesota Medical School.
I grew up in a small town on Long Island called Westbury, New York. At the time, it was a farming and whatever area for ducks and potatoes, so it was really an interesting environment. So I was in college at Columbia, and I was a experimental psych and anthro major, and what I was really interested in was neuroscience, but as a discipline it didn't exist then. So I took mostly graduate courses in my senior year, and then the decision was to go to medical school or graduate school, and I said, I think medical school is going to give me more options to do things when I was done. I went to medical school at State University in New York, in Syracuse. It was a great place to be a student, and we did lots of hands-on care throughout our rotations. When I was a third-year medical student, on one of my later rotations, I took care of a 6-month-old child that had leukemia in the CNS, and it was very troubling to me. And then on an elective a few months later, I actually did an autopsy on that child, and it really moved me to say, I think that a collective body of individuals doing research might be able to impact these things like I just saw with that child with leukemia. And pathology was the route to do that. The University of Minnesota and 2 other places in the United States only at that time, this is the early 70s, had outstanding what we call laboratory medicine or clinical pathology. One was where I was in Syracuse. Minnesota was probably the preeminent institution. The third was the University of Washington in Seattle, and that had been started by a former University of Minnesota faculty member. So I went to the best place I could, and fortunately they accepted me, and I've been very happy not only with my training but with my career. Very fortunate at the University of Minnesota. When I got interested in research as a 3rd and 4th year medical student, I started taking graduate courses in tumor viruses, among other things, and they so intrigued me, and I was passionate about that, and wound up working in a professor's laboratory part-time while still being a student. And it was, for me, the most exciting work I'd ever participated in. I said, this is what I think I want to make my career. As we know, cancer, if found in an early stage, is often curable. What really leads to the morbidity and mortality of patients is the spread of the cancer and metastasis. So I was very interested in the role of what we call extracellular matrix molecules in the spread of cancer cells. And also, these are in the wall of blood vessels, so if a tumor invades the bloodstream or elsewhere, it has to go through these molecules, which serve as a barrier, as we know, in a vessel. And looking at those dynamics, and then the fact that some of these molecules can actually promote the growth of tumor cells, I thought there was a big opportunity to not only do research, but actually look for potential targets for therapy. So I've been very fortunate that I've been asked to do various things along the way in my career that I had never intended being involved with. And at the time, the University of Minnesota had, on paper, an MD-PhD program, but it was hardly viable. It wasn't a fully integrated program. Essentially, you went to med school and graduate school, totally separate types of training. And the prominent institutions in the country have all prided themselves in a very strong MD-PhD program. So a group of leaders under the aegis of the dean came together. I was one of them. And as a physician scientist, was kind of like the prototype of what the program would hope to train. And I was asked to do this, and did it, and I really enjoyed it. It was like climbing Mount Everest, and people said we were too early to get grants, and I said, well, we're going to try anyway, because we didn't only had a 2-year track record, and we got the grant. So that training grant that funds, many students for the period of time they're in an MD, PhD, has been fully active for more than 30 years now, and I was the first PI. So that was very rewarding to me. Well, it's sort of like what led me to go into pathology in the first place, taking care of patients. There's a limited end that you can impact in any one day and whatever. And then in academic medicine, as a researcher yourself, you can do outstanding things, but it may be, very important research, but the impact that might have is very difficult to see in the near term for sure. So I felt if I got into some forms of academic administration, I would have more resources available that I could deploy and give to very talented people and see and watch their careers flourish. And that has happened over and over again in my career. For various reasons, I've been asked by leadership to take on responsibilities, or in fact, I've made the case that we needed to do things that weren't happening, and the dean and his team supported that. So in the case of the Biomedical Engineering Center, a substantial gift from Medtronic had been given to the university to develop a center and name it after one of the founders of Medtronic, Earl Bakken. And it laid pretty dormant for a number of years, and there was concern on Medtronic's part if we could really develop that. So what occurred was the dean of the School of Engineering at the time, the medical school, asked me to do this. I set it up. It was a tremendous success, and is now, the name has been changed, but it's extremely important with hundreds of faculty members of that. The Cancer Center was really led by John Kersey, and John and I had labs right next to each other, very close relationship. And basically what John and I did is probably for the first 5 or 10 new hires for the Cancer Center, they were in our department, lab medicine pathology. So we worked very synergistically, and it was a great opportunity. In regards to the Immunology Center, being a major transplant center here, I felt we really needed to have extremely strong research in immunology. A senior faculty member had left, and it presented an opportunity to hire new people. And I went to the dean at the time, Dave Brown, and said, I think we should start an immunology center and get other departments to buy in. They did, and we hired lab medicine hired the first 4 or 5 faculty members. It's been an outstanding success. The Immunology Center in Minnesota is known internationally in the highest regard. Very hard to imagine the evolution of our testing capabilities that's occurred during my career. When I was a medical student, finishing in the early 70s, we would see patients with sickle cell disease or these other hemoglobinopathies, and we would joke about someday we'll find a gene and somebody could correct these things. And nobody in their wildest imagination ever dreamed we'd be talking about, gene therapy and cell therapy today. The diagnostic capabilities we had in the clinical laboratories, they were okay, but they were very rudimentary by today's standards. We've gone up 2-3 logs in capability, probably, in the prowess of the laboratory diagnostics. For example, in microbiology today, many of the diagnoses are made by mass spec, a chemical assay. And no card-carrying microbiologists back in the day would ever imagine they would be doing anything with a mass spectrometer to do their work. And there's examples everywhere in DNA technology and many other things. So it's a brave new world we're in, and it's exciting, and it's why I enjoy coming to work every day, because things continue to move, and we bring in new young people who flourish in these new technologies, and it's very, very, very fulfilling for me to see this happen. FASEB is a confederation of more than 20 biomedical research organizations, and its main objective is to set and work on public policy and advocacy for the biomedical research community. They have now some 130,000 members, and this is an organization that actually Congress looks to for advice and recommendations on funding and various other things. So it works with the AAMC to deal with Congress on NIH budgets and funding for research, new initiatives, either in child health or in aging or HIV and what have you. So it was a real honor for me to be asked to be the president of that organization, and I had very close working relationships with the director of NIH. There was a move to try to reauthorize their budget, and great risk in that it might be cut. So I, with the director, and then worked with a congressman who was the head of the Energy and Commerce Committee of the House to work on this, and it was very, very rewarding. Also, it was a time of great tension in the country regarding stem cell research and other factors. So we would advocate for the rights of scientists and physician scientists to be able to do that type of research. There was also concern about the use of microorganisms like E. coli that could be used for dual use, and that is one for research, but also could be used as a weapon. And genetic engineering to make them behave very, very dangerously to humans. So one had to work through the ethics of all of that and how to appropriately deal with it, and we were at the table with all those high-level discussions. So I was fortunate with co-author Bill Hoffman to write two books, The Stem Cell Dilemma and The Biologist's Imagination. What the genesis of that goes back to my period of time working with FASEB in Washington and the great debates on stem cells. So what I saw was very well-intended people in the Congress and also in the advocacy community. I think at the time the most notable was the Juvenile Diabetes Foundation, but there were others who were clamoring to push for the use of embryonic stem cells for research. And what I observed was that most of the parties didn't know very much about what they were talking about. And I felt there was a need for something that would be addressed to the lay public, an educated lay public individual, who might, if you will, educate them and set the record straight on these things. And I think what many didn't know at the time was that these embryonic stem cells actually form tumors. Most of the time they're benign, but they can transform and be a malignancy. So the prospect of putting embryonic stem cells in a child to cure some disease, it was probably not ethical to really promote that. So anyway, the idea was to put this information out there and try to get a balanced view for the pros and the cons of using various stem cells and their potential utility as therapies. The diagnostic capabilities that we have today, that have even evolved in the last couple of years, are so profound and so important relative to not only the diagnosis, but then the potential therapy that people with, for example, cancer might have, that it's a whole new world available to us. The traditional pathology would look at a slide under a microscope and make a diagnosis. Then the next generation of capabilities was the use of antibodies that could target certain molecules in cells, and they would be attached to an enzyme with a dye, and color could form that was then visible in the microscope, and that was called immunoperoxidase. Now there are new capabilities with the opportunity to use not only genomics and screen cancer cells for various genes that are playing a role in the onset or the progression of that malignancy, but also there are technologies that we're just beginning to start here at the University of Minnesota in the first quarter with a very sophisticated machine that can look at cell tissues with up to 50 antigens at one time, using antibodies that are linked to heavy metals that you then measure by mass spectrometry. And I see a day, and not in the distant future, this is a research tool at the moment, but I see a day in the not distant future, certainly in my lifetime, that pathologists and clinicians will have not only the tissue selections and the diagnosis of this type of breast cancer or not, but they will also have the pathways that are perturbed or abnormal in those tumor cells, which will then direct you to what the most appropriate therapy would be for that patient. So that is personalized as personalized can be. And again, this is going to mean huge things to patients for not only the morbidity and decreasing mortality. I think it's going to be a wonderful thing for oncologists and surgeons to have these capabilities that really only pathology can provide.